Hello, I'm Robin Brumble, and I am the Senior Director of Scientific Affairs at CLL Society. We are entering into our 4th year since the start of our research program, for which just this short period of time, we've already committed to funding $1.7 million solely to CLL and SLL research. Today, I am joined by our 2025 Clinical Scholar Award recipient, Dr. Matt Davids. And this particular award is the largest in size that we offer annually, and we are extremely thankful to the many individual donors in our community who helped make this award possible. Our Clinical Scholar Award is specifically dedicated to an exceptional physician researcher who has already established themselves in the field of CLL, and it is designed to support the research efforts of an advanced physician scientist who is engaged in independent clinical research. And there is also priority given to someone who's demonstrated exceptional leadership, scholarship, and academic success. The overall goal of this particular award is intended to advance the development and treatment options for CLL. So Dr. Davids, at this time, I would like to first start off by saying congratulations. And if you could maybe begin telling us a little about yourself, and I would love to hear about what led you to the decision to dedicate your life's work to helping individuals with CLL and SLL.
Sure. So let me just start by saying a heartfelt thank you to the CLL Society for this award and for believing in me and my research and my team. It's really a huge honor to receive this award, and I'm incredibly grateful for the support. It's going to make a huge difference for the work that we're doing. So I grew up in the Boston area, so I haven't gone very far. And I've been at Dana-Farber now for about 17 years, starting with my fellowship training there. And around the time when I was trying to do my initial research as a fellow, I joined a lab that was focused on cell death. In other words, the process on how cells normally die. And around that time, there were sort of some new drugs that were just being tested initially in sort of preclinical models and then eventually in the clinic that were targeting this cell death pathway. And so I sort of had developed this expertise in the lab. Actually, my first interest was in lymphomas. And we were having some trouble in the lab getting samples from the lymphoma patients because every time we needed a new sample, we had to do a lymph node biopsy, which is an invasive procedure. And we kept getting samples in the lab from patients with CLL who were generously donating blood samples and occasionally bone marrow samples. And so I just started doing some of these experiments on cell death in CLL patient samples and started to find some interesting things. And when it came time for me to get a job as an attending physician at Dana-Farber, they actually needed a doctor focused on CLL. And I had been doing my research in the lab on CLL, so that was sort of a natural fit. And so I started seeing patients with CLL on a more regular basis. And even at that time, CLL patients were living for a few years, and you kind of develop these nice relationships with patients. And so for me, it really kind of blended the incredible science that was going on also with these strong patient relationships that we can develop over the years. And that also happened to be a time when the first drug targeting cell death came into the clinic, which you guys probably know now as venetoclax. And that was exactly the drug that I had been working with on the lab side for a few years at that point. And so when it came time for that clinical trial to open, I had the opportunity to lead the first in human trial of venetoclax at Dana-Farber. And we treated some of the very first patients in the world with the drug, which was called ABT-199 at that time. And that really helped kind of launch this whole era of targeted therapies with the BTK inhibitors like ibrutinib at the time. And we were working with PI3 kinase inhibitors like idelalisib. And I feel very fortunate that I was sort of at the right place at the right time, where I was able to then start combining these drugs in the lab side and developing new combination therapies. And now, of course, we're seeing all the benefits of that for patients with CLL living a very long time with these drugs that are both more effective and better tolerated than the old chemotherapy drugs.
Great. That's wonderful. I learned something new about you today, so that's good. Well, your research project is called Optimizing Glofitamab, and please correct me if I'm not saying that correctly. Optimizing Glofitamab Combination Therapy in Richter transformation. So I guess let's talk a little bit about Richter transformation first. What exactly is this condition and why it's important to study it in relation to CLL and SLL?
So, over the course of my career in CLL, we've seen these dramatic improvements in the outcomes for most patients with CLL, with these targeted drugs like BTK inhibitors and venetoclax. A lot of patients are doing really well. But unfortunately, occasionally patients can have a transformation of their CLL, which means that these kind of normally slower moving cells can transform into more aggressive cells, usually a type of lymphoma called diffuse large B-cell lymphoma, which actually, when diffuse large B-cell lymphoma arises in someone spontaneously, it's usually a very curable condition. But for reasons that historically we have not understood very well, when DLBCL like this arises in a CLL patient, the outcomes are historically not as good. It's much harder to treat this. And we've found, unfortunately, over the last few years, despite all the advances with the new drugs that we have, patients with Richter's transformation are really not doing well still. So this has really become a major area of focus for my own research and several other researchers in the field to really try to focus on this group of patients with Richter transformation to try to improve outcomes. Now, sort of the traditional approach to treating Richter's transformation has been to use chemotherapy. And historically, that did lead to a complete remission in about 20% or so of patients. So again, not great, but there was some benefit there. Over the last several years, we've been leading some work where we're combining venetoclax with chemotherapy, and actually just recently presented the update of these data at a meeting in Europe. And we're seeing about half of patients achieving a complete response with this approach. So it does seem better than the chemotherapy on its own, but certainly does leave a lot of room for improvement. And improvement not just in terms of how effective it is, but also some of the side effects, because as we know, CLL patients are often older and have other medical issues, and giving intensive chemotherapy is not so easy. There's a lot of side effects. And so that sort of led to some of the initial discussions about, could we develop a regimen to treat Richter's transformation that used a non-chemotherapy based approach? And we'll talk, I think, a little bit more about what glofitamab is, but there's a number of different approaches now being studied, and we chose to pursue glofitamab because we think it's a very promising way to treat Richter's.
Great, yeah, so let's briefly touch on glofitamab. It's not a drug that's very common for those with CLL, so can you maybe touch on what kind of drug it is, how it works, and what we know up to this point in time about its use?
So glofitamab is what's called a bispecific antibody. So CLL patients may be more familiar with what we would call monoclonal antibodies, which if you study the roots of words, mono, of course, meaning one. So most of these antibodies like rituximab or obinutuzumab that you may be more familiar with, they target one thing on the cell, usually a protein called CD20, which is specific to the CLL cell. So that's why these drugs are effective in the disease. But what a bispecific antibody does, as you might be able to guess, actually targets two different things. So it also usually targets CD20 on the CLL cells, but it also targets a protein called CD3, which is on T cells. Now, T cells are kind of a normal part of your immune system that's normally there to help you fight off infections. But what these bispecific antibodies do is that they actually train your normal T cells to actually attack the CLL cells. They actually grab them and they kind of bring them into closer proximity so that your own T cells can kill off the CLL cells. Sounds like a little bit of science fiction almost, but it actually seems to be showing some really good promise in both CLL and Richter's. In fact, for those of you who follow Dr. Kaufman's story, he's been treated with epcoritamab, which is also a bispecific antibody that's showing promise in this disease. So we had been aware that there was kind of a large lymphoma study with glofitamab that had shown a signal of some complete responses in a very small number of patients with Richter transformation. And so we decided to ask this question, could we do a larger kind of proper study where we look at a large group of patients with Richter transformation and see how effective glofitamab is? And that really forms the nucleus of sort of the first part of the grant that I proposed here. But then, we're going to be looking at combinations as well. And I think we'll get into that as we kind of proceed with our discussion here.
Right, yes. So yeah, my understanding is your research, like you said, is it's not just looking at the glofitamab as a standalone treatment. You're going to look at, like, as you said, the effects of combining it with other drugs. I think maybe pirtobrutinib is one and a couple others. Can you talk about that a little bit?
Yeah, so we always try to be very systematic when we're studying these drugs because what if glofitamab actually were incredibly effective on its own? We wouldn't want to not study that, right? Because that could be really good for some patients. But as we've learned over the years with Richter's transformation, this is a really challenging situation, and it's unlikely that glofitamab on its own is going to help all the patients. And so that's why from the start here, we're planning to do various combinations as well. So the study's starting with a group of patients treated with glofitamab on its own. And then after we finish that initial part, we're going to open 3 different, what we call cohorts of patients, basically just groups of patients who are treated with these 3 different combination partners. So probably the one that CLL patients are most familiar with is pirtobrutinib. And this is what we call a non-covalent BTK inhibitor. It's just kind of a newer type of BTK inhibitor that can work even when the older ones like ibrutinib or acalabrutinib or zanubrutinib have stopped working. And so we think that this will have a couple of potential benefits. One is that the pirtobrutinib can control some CLL if it's still around. But also pirtobrutinib on its own has been shown to cause a response in about half of patients with Richter's transformation. And so combining these two together makes a lot of sense. As many of you know, pirtobrutinib is generally well tolerated. It does not have a lot of side effects. There's even some indication, and we're going to study this, whether pirtobrutinib might actually help to lessen some of the side effects that we can see with glofitamab. Glofitamab can cause some immune reactions, something we call cytokine release syndrome. And there's some, at least preclinical evidence suggesting that drugs like BTK inhibitors can help to reduce that type of inflammation from bispecific antibodies. So that's one of the cohorts. The other two cohorts may be a little bit less familiar to patients with CLL because these are drugs that we tend to use more with aggressive lymphomas. One of them is called polatuzumab. This is a really interesting drug. So it's called an antibody drug conjugate, or ADC. It's basically an antibody that sticking off the end of it has a chemotherapy molecule. And so we know when we give chemotherapy to a patient, generally there can be a lot of side effects, but what's amazing about this antibody is it can selectively deliver the chemotherapy only to the tumor cells and spare the other cells in the body. And just recently, this polatuzumab drug has been FDA approved in combination with a standard regimen that's been given for 25, 30 years now for other lymphomas like diffuse large B-cell lymphoma, and it started to become part of the standard of care. So because it's so active in the regular diffuse large B-cell lymphoma, we're going to explore how active it is with glofitamab in this Richter's population. And then the final combination cohort is very interesting as well. It's using a drug called atezolizumab. This is a monoclonal antibody, so targeting one thing, and it targets sort of what we call the molecular camouflage that can be on CLL cells. So sometimes CLL cells can put up this camouflage so that the immune system can't see it. So even if that glofitamab is having that effect on the T cells, it can't really easily find the CLL cells because of the camouflage. And so the atezolizumab pulls back the camouflage and reveals the CLL cell to the antibody. And again, sort of from the preclinical side, from the lab side, seems like this would be a really logical combination partner. It may have more side effects as well in terms of immune stimulation. So we need to make sure we're getting the dose right and that it's properly safe. And this is the type of thing we'll be exploring in the study. So we're really equally excited about all four different cohorts, the monotherapy with just the glofitamab, and then each of the three combination cohorts. It's really hard to know ahead of time which of these might be the most effective. It's possible they're all effective in different ways and for different patients. And that's going to be one of the focuses of the grant, is kind of figuring out, are there certain patients who would be better suited for one of these combination partners versus another? And are there certain patients who might do just fine with just the glofitamab and might not even need the combination partner?
That's fascinating. Well, you kind of already touched on a little bit of the methodology, but I know one question we always get from our community is, well, who's going to be eligible for this study? So maybe talk a little bit more granular on that level about methodologies that you'll be using.
Yeah, so for the trial itself, we tried to keep the eligibility as broad as possible. So there does need to be a confirmed diagnosis of Richter transformation, but we don't have any particular restrictions around whether patients had prior treatment for that or not. Like some studies are restricted only to patients who had prior treatment for Richter's and some not, but our study will allow all patients. Similarly, a lot of patients with CLL have a history of being on a BTK inhibitor drug. That may affect sort of which cohort they wind up in, but it doesn't affect their eligibility for the study. They'd still be eligible. So there's some nuances in terms of which patients get assigned to which group of combination partners. But we really tried to be as inclusive as possible to allow as many patients with Richter's transformation to enroll. And so, I think the other real main focus of the study is understanding some of the biology of what's going on when we're using these drugs. And so we're using a variety of different lab techniques. A lot of this is focused on the different immune cells that are present. And one of the technologies that we're planning to incorporate is called CyTOF. And I'll explain sort of what that is, because, a lot of people are not familiar with this, but one of the main tools that we've been using historically to study CLL and Richter's is something called flow cytometry. And this is a way that we can look at different proteins on the surface of the cells. Like I mentioned CD20 before, that's how we know that CD20 is there. We can run these cells through a flow cytometry machine, and we can see the protein on the surface of the cells. The problem is that flow cytometry is pretty limited. We can usually see about 6, maybe 8 markers on the cells at most, and that really kind of limits our understanding of the biology. So this newer technology, CyTOF, uses a different way to look at the cell surface proteins, and we can look up to 30 or sometimes up to 40 different markers on the surface. So it's many-fold more in terms of the biology that we can understand about the different populations of immune cells. And we're going to do a lot of different sequencing techniques as well, sequencing RNA, sequencing DNA, really dissecting out the biology here as best we can. And importantly, this will be on the samples from the patients on the trial. And the reason that's important is because once we have the scientific data, we can then go back and look and see, how did those patients do on the therapy? And can we find patterns in the immunology to predict, these are patients who we might want to go with this combination because they're more likely to respond. And maybe these patients are more likely to respond to a different combination or to the single drug, or maybe there's patients who we can predict are not going to respond to this type of therapy at all, and maybe they need to go back to the more traditional chemotherapy. So the study's not going to necessarily definitively answer all those questions, but a lot of it is kind of taking those first steps toward a more personalized approach to treatment. And we hope this is really a foundation that's going to lead to future studies as well.
Great. Well, I think that probably answers the question of what you hope to ultimately accomplish, which sounds like a lot. Is there anything else that you could think of that if you could tell someone what is the biggest thing that you hope to accomplish with this study, what would that be?
Yeah, I mean, the big goal is to improve outcomes for patients with Richter's, ideally to develop a cure for Richter's. We think it's possible with a subset of these patients. We're not going to cure everyone, although we'll try. Historically, if you look back, the only type of treatment that's been able to cure patients with Richter's is a donor stem cell transplantation, like a bone marrow transplant from another person, which has so many side effects and risks, and it's not something that a lot of CLL patients are candidates for. So one reason why I'm particularly excited about this study is that it's also an immune-based study. It's, as I said before, it's using your own immune system. And so it gets around a lot of those risks of the donor stem cell transplant, and yet also has that potential benefit for the long-lasting immunity that we think can lead to cure in a subset of patients. So there's that aspect that makes me excited. The other thing I would say is that we always try to work with drugs that are already approved, because it's difficult to get a drug approved just for Richter's. It's a relatively small population. So all the drugs that we're using in this trial are already FDA approved. And that's really important because it means that if the study is positive and we find some really good results, that could be applied to patients with CLL potentially right away. And I think a good example of that is the recent work that we just did with the chemotherapy with venetoclax that I mentioned earlier. Based on our study, that's now been listed in the NCCN guidelines, which is basically the recommendations that experts give to oncologists around the country. And when things are listed in those NCCN guidelines, often insurance companies will pay for them. Actually, at this meeting that I was just at presenting in Switzerland, we presented these results, and already I've had people from around the world contacting me asking for advice about their patients because venetoclax and the chemotherapy are drugs that are available in other countries. And so the impact that we have with these types of studies is not even just here in the US, but it can be worldwide. So that would be sort of my bigger aspiration for this glofitamab trial, because this is also a drug that's approved in several countries now around the world, is that what we discover here with the generous funding from the CLL Society is something that we could then apply to CLL patients who develop Richter's around the world. And that would be great if we can really make an impact for the global community.
Yeah, that would be really great. Well, do you have any other final thoughts that you would like to share as we close?
Again, just to close kind of where I started, which is that I'm incredibly grateful for this award. I've worked closely with the CLL Society for many years now, particularly with Dr. Kaufman, who I think I first met maybe about 15 years ago at this point, when we were both a little bit younger and a little bit less gray. But it's amazing to see how well that he's done by kind of benefiting from all the great therapies that have been developed for CLL. And we really are grateful for the support from the CLL Society. It's really making such a difference, not just with the research program, which is huge, but also for patients around the country and around the world as a resource for education and support. And so keep up the great work with everything you guys are doing.
Thank you. Well, Dr. Davids, on behalf of CLL Society, I'd like to thank you for all of the work that you are doing to advance the research in CLL and SLL. And we are all very eager to learn about your future findings. And I think I can speak on behalf of both CLL Society and the community that we serve in saying that we're all very much looking forward to hearing more in the future about the results of this study. So thank you so much for your time today.
Thanks, Robin.